The next speaker is David Atwell, who's going to present on parasites in neurovascular coupling in health and disease. And we'll have the discussion at the end of the presentation.
Can we have the lights down, please? These lights. Merci pour l'invitation, Marie-Germain. Today I'm going to talk about regulation of brain energy supply at the small vessel level, and I'm going to talk about where the action begins and where it ends. So specifically, I'm going to address the question of what? This is not good. Did you change something back there? Okay. Try again. So we're going to... What I want to do is to, first of all, look at where brain energy supply is controlled, whether that happens predominantly at arterioles, as is the dogma, or in capillaries. I'll show you that parasites can dilate capillaries. I'll show you how fast it can work, and then look at the relevance of this to bold imaging. And I'll then switch to pathology and look at the effect of ischemia on capillaries and the role of parasites in the loss of control of brain energy supply in pathology. So here's the plumbing of the brain in rather simplified terms. Blood comes in through penetrating arterioles, and the dogma is it's the smooth muscle around these penetrating arterioles, which is the primary site of control of brain energy supply. But that's not the whole situation, because there are also these small, isolated contractile cells, which Costantino just told you about, which were discovered by our hero, Charles Rouget, back in 1873. Now, Charles Rouget was a bit of an outcast in the French medical establishment. He couldn't get a job in Paris, and poor man, he had to live most of his career on the south coast of France. Eventually, he got a job in Paris. But after he discovered parasites, pretty much nothing happened for about 100 years. This is the number of papers in PubMed with parasite in them. But now the number has taken off exponentially, and you're going to be hearing lots more about them. And for the CADASIL community, it's relevant that parasites express Notch3, just like smooth muscle cells do. This is what they look like. Here looks a bit dim here. Here we've labeled parasites on retinal capillaries or cerebellar capillaries using an antibody to a proteoglycan that they express called NG2. And if you look in close, you can see they put out long processes along vessels, and if you patch clamp them, you can see they put processes around vessels, so they seem designed to kind of cuddle the capillary and produce a spatially localized constriction. And Edith Hamel's group has shown that 2/3 of the noradrenergic innervation of the brain ends near capillaries rather than arterioles. So maybe that's telling us something about where amines might be controlling blood flow. So what do? Well, about 10 years ago, we did a very simple experiment here shown on, I'll show you a movie of this in a minute, on retinal capillaries, where we pushed an electrode up against a parasite and depolarized it, and we were amazed to find it pinched off the capillary completely. You can see the parasite here, and this is recoverable, reversible. Here you see the diameter decreasing almost to zero and then recovering. And this only occurs where there are parasites. If you stimulate a place on the vessel where there aren't any parasites, there's no constriction. And it's mediated by calcium. So here's a movie of this happening. So what I'm going to show you, I'll play this a couple of times, is there's a parasite under here we're going to depolarize, and I'll show you that it constricts the vessel. But as Costantino just intimated, signals travel along vessels, and this doesn't just occur at the arterial level. Parasites talk to each other as well, so after this one constricts, you'll see this one constricts a bit later, but there's no change in the intervening capillary. So if I play it, you see this constricts, and then a bit later, this one constricts. So information hops from one pericyte to another along the capillary, perhaps going through endothelial cells, as has been shown in muscle, or perhaps there are gap junctions between the processes of the capillary. I'll just play it one more time. So this one constricts, and then this one constricts, and it goes in both directions as well. And if you plot out the diameter and you do this repeatedly, this is the repeated constriction of the middle one, and you can see the pericytes on each side repeatedly constrict, but the capillary in between doesn't change diameter. So what's the use of that? Well, it turns out that neurons are closer to pericytes, of course, than they are to arterioles. This has been quantified in the hippocampus by Thelma Lovick. They're 5 to 10 times closer. So you might expect if they want to adjust their energy supply, they would signal first to pericytes, and that signal might then propagate back from one pericyte to another. I've shown you this for constrictions, but there is some evidence in the literature this happens for dilations as well, from Don Puro's group. And then perhaps onto the arterial smooth muscle. Now, I've shown you them being constricted by putting a big electrode up against them, but luckily it turns out they can also be constricted or dilated by messengers released from neurons and glia, and I'll show you that in a minute. And we want to understand this to understand normal control of blood flow, altered blood flow in pathology, and also functional imaging signals, which the scientific establishment pours squillions of dollars into each year, but nobody really knows what's being imaged. So how do these cells respond to transmitters? We've looked at this first of all in brain slices. I'll come to in vivo data later. And we've either applied noradrenaline to mimic endogenous noradrenaline release from the locus coeruleus, or we've superimposed on that glutamate to mimic neuronal activity. This just shows you what I'm going to show you in a movie in a minute. Noradrenaline constricts capillaries. The mean diameter of the vessels in these studies was about 4 microns. And superimposed glutamate dilates them. So here's a little movie showing this same thing. So this is in the cerebellar molecular layer, and there's a pericyte just here. And when we apply noradrenaline, you'll see that this vessel constricts just here, and then if we superimpose glutamate, it dilates again. Okay, so these pericytes can produce localized constrictions and dilations in response to neurotransmitters. So we want to know how fast they can do that, in vivo especially, and what's the relevance to functional imaging. So let's just get functional imaging out of the way first. If a bit of the brain, like the cerebellum here, wants to be more active, then it needs more energy, which of course comes in the form of oxygen and glucose in the blood. So you would think then that more oxygen would be used, and that would increase the concentration of deoxyhemoglobin. Now, deoxyhemoglobin, it turns out, is like a little magnet. It's paramagnetic, and it decreases magnetic resonance imaging signals from the protons, which is what is imaged in those experiments. So it messes up the magnetic field, and it turns out that means that the protons, which are spinning, precessing around a superimposed large magnetic field, they get more out of phase, and so the signal gets smaller. So you might naively expect that when you use more oxygen, there'd be more deoxyhemoglobin, and that would decrease functional magnetic resonance signals. But it turns out that, of course, as Costantino has said, signals go from the neurons over to the vasculature, and these are predominantly mediated by glutamate, which releases a series of vasoactive messengers, and that leads to more blood flow occurring. And so as a result of that, there's actually a decrease of the concentration of deoxyhemoglobin because too much fresh blood is coming in. And this decrease in deoxyhemoglobin actually increases the functional imaging signal, and that's what you see as these little red dots on these lovely pictures, which people regularly publish in high-profile journals. So what are they imaging? Well, mostly they're imaging the flow increase. There are some sophisticated, some little technical things we don't need to worry about. They're basically measuring the flow increase. So where is that going to be initiated? Well, just from the point of view of a design approach, here's the vasculature again with the peel surface, the penetrating arteriole, and then the blood going through increasing orders of capillaries. If you've got some active neurons and astrocytes, which might release vasoactive messengers as well, then because these cells are closer to capillaries than they are to arterioles, we would expect these dilators released by the active cells, which are 10 times closer to the capillaries, to reach the capillaries first. Now, I don't have time to go through the experiments we've done looking at what messengers mediate these dilations of pericytes, so I'm just going to summarize the results. As Costantino said, there are many messengers involved, but we found that it's predominantly arachidonic acid derivatives which are relevant for the pericytes. And the main one that's relevant is prostaglandin release, which acts through EP4 receptors. So all the dilations I'm going to show you are blocked by a blocker of EP4 receptors. But that's not the only thing that's happening. You also need nitric oxide to be released in order to suppress the synthesis of another arachidonic acid derivative called 20-HEAT, which would otherwise constrict the pericytes. Okay, so the dilations are done by prostaglandins. Now, how might that work? Well, if we patch clamp these pericytes, this is in a mouse where DS red is expressed in the pericytes, so we can identify them. We can then patch clamp them and fill them with Lucifer yellow. If we apply glutamate or NMDA, we get an outward current produced, which will hyperpolarize the cells and decrease voltage-gated calcium influx. And the same occurs if you stimulate endogenous glutamate release by activating the parallel fibers in the cerebellum. Again, you get an outward current produced, which will hyperpolarize them, and you would expect that to relax them. And this is indeed what happens. If you image these capillaries in this case in the cerebellum, and activate the parallel fibers, after you've pre-constricted them, in this case we use noradrenaline to mimic noradrenaline release from the locus coeruleus, which would be happening in vivo, then each time you stimulate them, you get a little dilation of the capillaries. Okay, so this is all in brain slices. In vivo, people don't like work in brain slices because there's no blood pressure and there's no flow, and that might alter messengers released from the endothelium, for example. So what about in vivo? To look at this, we've teamed up with Martin Lauritsen, one of our Leduc collaborators in Copenhagen, and we've been interested in which dilates first. The capillaries, as I suggested, ought to be the case from a design point of view, or the arterioles coming down here from the pia into the brain. And so we use two-photon imaging in vivo to look at the diameter of the penetrating arterioles and the capillaries and just see which one dilates first when we stimulate the whisker pad and look in somatosensory cortex. This is exceedingly dim. Maybe you could put the lights out completely. Thanks. Anyway, this is looking down on the somatosensory cortex. This is a penetrating arteriole just here. It's very dim. This is the first order capillary coming off it, and you may be able to see the red pericyte superimposed on it. If you do brief stimulation of the whisker pad, you get a dilation of the vessels, which occurs in a second or so, just as is seen for functional imaging experiments. We actually use slightly longer stimuli to get a bigger dilation, which is easier to measure. And the first thing I want to show you is that all vessels dilate. Okay, so this is what we call the branching order. So 0 is the penetrating arteriole, 1 is the primary capillary coming off it. This has no continuous rings of smooth muscle, it just has isolated pericytes on it. And these are the higher order capillaries. And roughly about 40% of all vessels dilate. Now, the fact that the capillaries dilate doesn't mean it's an active dilation. They could be being pushed open by the increase of blood pressure that's generated by the arterioles dilating. But in fact, I'll show you it is active in a second. So what I want to show you is that, let's just see if I can get this. Nope, I'm just going to open it again then. What I'm going to show you is a movie of imaging simultaneously the penetrating arteriole, which is shown just here, and the first order capillary, which is shown just here, and you may or may not be able to see the red pericyte on each side of it. Great. This is not what I want. So when I play the movie, the bottom here is the time course of dilation of the first order capillary and the penetrating arteriole, but I'm going to play it several times because the dilations are small and I'm not sure how easily you're going to be able to see them. So I just want you to focus on this capillary first of all. when I play the movie. So when I play it, okay, I need to be more coordinated. I'm going to play it now, and you should see a small dilation of the capillary when I play it. So now, you see the capillary dilating? I'll do it again. Okay. Now. Right. In fact, it dilates when the line passes this increase here. So this is the last time to look at the capillary. Now. Okay, and now I want you to look at the penetrating arteriole, which is over here on the right. And I'll play it, and you should be able to see it dilate now. Okay. And if you look at the relative timing of them, I'll do it one more time. Okay, the capillary dilates and then the arteriole dilates. And in this example, the arteriole dilates about 3 seconds behind the capillary. Okay, so now I've lost control of the presentation. Okay. So that's what I've just shown you. And if you average over all the vessels that we've simultaneously imaged like that, then on average, the penetrating arteriole dilates about a second after the capillaries, which is what you'd expect if the dilating messengers get to the capillaries first, and there is active dilation of the pericytes. It's hard to explain the capillary dilating first if it's a passive response to the arteriole dilating. And in fact, if you do the quantification, you find that there's about a 1.3 second delay comparing first-order capillaries with north-order arterioles. The arterioles dilate about 1.3 seconds after the capillary. Whereas if you compare higher-order vessels, second-order capillaries with first-order capillaries, and so on, there's no significant difference. So all the capillaries dilate at about the same time, and they all do it before the arterioles. And that's shown where now, instead of simultaneously imaging, we're just averaging over all the vessels that we've imaged. And you find that the first and second order capillaries dilate, on average, 1 second before the arterioles do. Okay, so the fact that this dilation occurs first in capillaries suggests it's an active dilation. But there's another piece of evidence supporting that, which is that if you look on the capillaries at places where there are pericytes and places where there aren't pericytes, then you see more dilations where there are pericytes, and we're defining the occurrence of a dilation to be more than a 5% dilation. So the occurrence is twice as often, and if you plot the probability distribution of the dilations, again, it's larger near pericytes. So again, this suggests that pericytes are actively dilating, not just responding passively. So here's a summary of our data. It looks a bit complicated, but the only point I want to get across is that all vessels dilate by roughly the same amount. On average, capillaries dilate slightly more than arterioles. So what does this mean for the control of blood flow? Well, David Kleinfeld has published this beautiful so-called angiome picture quantifying the diameter of all the vessels in a cortical column, more or less, and he's worked out the resistance for a typical pathway coming down from the pia to a layer 4 arteriole, and then going through the network of capillaries, and then going back up the penetrating venule to the surface. And he finds that the majority of the resistance in that pathway is in the capillary bed, not in the arterioles or the venules. So if you put in our numbers for the dilation seen in the different kinds of vessels, we actually predict that the capillary dilation generates 84% of the decrease of resistance, the increase of blood flow, at least within the parenchyma. So if you assume that blood flow is held constant at the peel surface, then the diameter changes we see would imply that 84% of the increase of blood flow is generated by capillaries. So we actually think that capillaries contribute to blood flow increases in 2 ways. First of all, the messengers reach them first, so they dilate first. And that pericyte hyperpolarization may then spread along the vessel and back to arterioles. So there's a kinetics part to what they do. They dilate first. But secondly, the size of dilation that occurs and the fraction of resistance which is in the capillaries means that they dominate the decrease of resistance which occurs within the brain parenchyma. So to summarize that bit, both arterioles and capillaries dilate, but pericytes can dilate capillaries actively via prostaglandin release. This works faster than the dilation of arterioles. And what this means is that it's pericytes which initiate the bold signal. So those little red spots are imaging flow increases generated by pericytes. Now I want to turn to the bad side of all this control of blood flow by pericytes at capillaries and look at the effects of ischemia. Back in the 60s, Adelbert Ames showed that if you perfuse India ink into the blood, then it goes through all the blood vessels in the cortex. But if you then mimic a stroke by cutting off the blood supply to a part of the cortex, and then let it back again afterwards to mimic removal of a clot using TPA or surgically or just endogenous clot-busting mechanisms, then the India ink no longer goes through all the vessels in the cortex. It's going into these ones, but there are big areas that aren't very well perfused. This is the no reflow phenomenon. Now, 10 years ago, we found that if we imaged pericytes in the retina and made them ischemic, then there's a contraction of the pericytes. They constrict capillaries. And the reason for that is shown here. Normally, you need to maintain a low calcium concentration within your pericyte, and that's done by an array of iron pumps, powered ultimately by ATP. So when you cut off the ATP supply, these pumps stop pumping, the calcium rises, and that activates the myofilaments in the pericyte. And so this little arm around the capillary squeezes the capillary like this, and that causes the decrease of the diameter of the capillary. Now, Turgay Dalkara showed that if you do MCAO on rats, he claims that pericyte contraction produced by oxidative stress produces a long-lasting constriction of capillaries. So before ischemia, horseradish peroxidase can, of course, go all through the capillaries, as can red blood cells. But if you image the capillaries after ischemia, then you find that there are bits of the capillaries which are pinched off, which neither the HRP nor the red blood cells can get to. And it's known from earlier work that if you mimic a stroke, then you cut off the blood supply. When you let it back again, after a period of reactive hyperemia, there's a long-lasting decrease of blood flow which occurs. We wanted to know why this constriction of blood vessels and reduction of blood flow was so long-lasting. And it turns out that pericytes are extremely susceptible to ischemia. This is using NG2 to outline the vessel and the pericyte just here. This is in control conditions, and this is after ischemia. And both of these slices have had propidium iodide applied to them. So the red cell here is a dead pericyte. Its membrane has lost its integrity, and PI can go in and label the genetic material. And an hour's ischemia kills about 90% of the pericytes. They're much more susceptible to fast death than other cells are in the brain. And as I'll show you in a minute, this also occurs in vivo. So we think what's happening is that after the calcium's gone up in these cells, it just overwhelms the cell and triggers their death. So what we conceive then is that the pericytes initially constrict because the calcium has gone up, but they then die in rigor, like rigor mortis. So they die constricted around the capillaries, and that produces a long-lasting constriction. So why do they die? Well, blocking action potentials has no effect, but if you block glutamate receptors, either AMPA and kainate receptors or NMDA receptors, then you can roughly halve the amount of death which occurs. If you do, this is a strong insult with glycolysis and oxidative phosphorylation blocked, but if you do OGD, oxygen and glucose deprivation, and then reperfuse, then what you find is that although there's quite a lot of death during an hour's OGD, the amount of death increases with reperfusion time. Okay, so a lot occurs during the OGD, but there's increasing death afterwards. And we imagined this might be due to oxidative stress because of the Dalkara work showing that the constriction seemed to be produced by that. But it turns out it really isn't. So again, with OGD and reperfusion, you can reduce the amount of death which occurs using glutamate receptor blockers or taking away calcium, but blocking nitric oxide synthesis has no effect, and neither does applying various scavengers of reactive oxygen species. So at the moment, we don't know why they die when the oxygen comes back again. We've done some experiments in collaboration with Alistair Buchan in Oxford, looking at MCAO in vivo, and again we find that that MCAO again produces an increase in the number of dead parasites at a time when there's very little death of endothelial cells. So parasites die at a time when endothelial cells are unaffected. So going back to this diagram of the long-lasting decrease of blood flow which occurs after stroke, we wonder whether this long-lasting constriction might reflect the constriction and then the death of parasites in rigor. So this suggests that parasites are a therapeutic target in stroke and maybe in other diseases. So what I've shown you today then, apart from the fact that capillaries are the prime initiator of increases of blood flow in response to neuronal activity in, both in brain slices and in vivo, in pathology, parasites constrict and die, and it seems that this role of parasites, they play a crucial role in the loss of control of brain energy supply in pathology. And although I haven't talked about it today, you need to have parasites functioning to have an intact blood-brain barrier. So the fact that they die after ischemia is likely to lead to further problems due to loss of the integrity of the blood-brain barrier. These are the people who did the work. This was the original work done 10 years ago. The messenger systems and the work in slices were done by the people in the middle row. Martin Lauritsen and Bodil Gesslein did the in vivo experiments which we analyzed and for physiology, and the pathology was done in Alistair Buchan's lab by Brud Sutherland. So my final take-home message is that brain energy supply, where does the action begin and also end in pathology? It does so at capillaries. Thank you.